It is my pleasure to introduce Angela, who is the Director of Bioinformatics at Labcorp. I had the pleasure of meeting Angela in Miami last year. We were on a panel together and she gave a really nice presentation. I said, I have this meeting coming up and I think what you will present is going to be very interesting and informative to the community. And she graciously accepted, so thank you. She doesn't work with Zifo. This is just one of the facts I want to bring up because this is not about Zifo, it's about us all working together and learning from each other. So let's continue the dialogue during the breaks and lunch and so forth and the reception tonight. Anyway, Angela is the Director of Bioinformatics at Labcorp. She's been at the company now for 16 years. She's an IT lead, and she's responsible for crunching large volumes of data, large volumes of diverse data to aid in developing new genetics tests, and also works on the LIMS system to support the data generation. So without further ado, thank you, Angela.
Thank you guys for coming today and letting me come as well. Like Margaret said, we just met recently, but our love of science became clear right away and we hit it off. So I'm happy I get to share that with you today. As Margaret said, I work in a high-throughput clinical lab, so whatever volume you can imagine, it's probably bigger. And then with that becomes bigger problems and lots of lessons learned. So I figured that I could go ahead and share some of those with you today. I'm obsessed with the life of a specimen in my company. The life is long, so I'm only going to talk about a part of it today because Margaret only gave me 30 minutes this time. But it starts when you're at your doctor, or now we even had at-home kits, so sometimes you can decide on your own that you need to order that test. And then you come into a lab and you get a draw site, or you might spit into a tube at home as we keep expanding our specimen types. And then at some point in your journey, you go from a patient to a specimen. And that's where you actually get packaged up, you get sent to our lab, we accession you, and then we go ahead and test you, we get some results, we send you a report and a bill. I'm only going to talk about this one little section right here. It's a long journey, like I said, but we're going to focus mostly on you've already been accessioned into our system and what happens to you, all the way down to the results that come out the other side and how we trend those. If I drilled into that red box, and kind of exploded it out for you. This is, in general, what the testing workflow looks like. It varies based on technology and test. We have the dry specimen workflow up top, so things like intake review, where a genetic counselor might be checking that your physician actually ordered the right test in the family. Maybe there's some info that you need to manually enter in as well. You might go through a prior auth step to make sure insurance is going to reimburse. And then depending on where you live, you might go through informed consent, right? So if you're in New York State or you're getting tested in Massachusetts, then you're going to have that extra step as well. And then some of those are gating factors into getting into the wet specimen workflow. We'll still go ahead and extract you because we want to have the highest quality DNA, but we're going to hold until you get through that dry lab step. On the wet side, obviously there's extraction. It also includes a quant, right? I'm a math person, so I love numbers. That's the first time I get a number that I get to play with. And then it's also about instructions for the robot and labels, and you'll see that one carry through all these boxes down below. When I say robots, I could be talking about Tecans, Kingfishers, Hamiltons. My team supports multiple sites, as does this LIMS, and you don't get to control the instrumentation that exists at that site. You just have to embrace it. On the assay prep side, we're building plates. Again, we're talking to the robots from the LIMS. We're creating instrument work lists so we can control what the settings are on that particular machine to a guaranteed high throughput. And then we get into the analysis. I'm going to drill down into this box a little bit further next, but this is really my cup of tea. It's everything from file orchestration. So how does that file get off the instrument, into the LIMS, back out the other side with no human intervention because it's probably at 2 a.m. when it's moving. And then all of the visualizations that come with the bioinformatics and those algorithms. And then we jump into interpretation, and that word is tricky because it means so many things to so many people. I'm going to use it in the context of variant scoring, assessing how a batch looked, QC metrics, and both that tech and director approval step. So I'm using a very broad definition. I'm sorry if that upsets anybody clinically. On the confirm side of things, again, there's a lot of different confirms. Sanger, MLPA, digital MLPA now, qPCR. low coverage genome. So the LIMS goes ahead and supports all of those as well as informatics. And then we finally get into this step called result processing, which is basically like the glue that ties everything together, funnel all the results back to one screen. The layout of that screen is key because nobody wants to side scroll or vertical scroll. So you need to figure out how to get 2 weeks of an experience for that specimen into one screen. And then the director's going to go ahead and review that, and you're going to shoot out the door, maybe even through auto release for reporting. So when I say testing, I just don't mean the test on the instrument. I mean this entire diagram here. So now let's drill down one layer further. In that analysis box, there was bioinformatics. And you might say, what is bioinformatics? I tell my daughter, I wear many hats at work. We're going to do reverse family feud. So what are the things you don't think of first when you think of bioinformatics? I'm in charge of annotations. That includes variant scoring. We do a lot of plotting. I spent my entire month of April making box plots. It was a lot. I made a dashboard now that makes box plots. You'll see it later. We do a lot of data mining. So you get lots of questions about, hey, that assay that ran 12 years ago and that LIMS that nobody knows about, I need to query some data out of it. And you're like, great. I'll write that query. Like I said, dashboard's my new favorite thing. We do a lot of automation. I have this file. I need to get it off this instrument. I need to get it through this software. I need to get it back out the other side. We get asked to help a lot with that one. So maybe those are all the things that you don't think of when I say I work for bioinformatics. What do you think of, or should you think of, variant detection? It's a decade of my life right there. Structural variants, CNVs, I've done them all. We focus a lot on technically complex regions, right? Can you get NGS to do things like alpha-thal or CYP21A2 or PMS2 if you're in the inherited cancer space, right? Those tricky genes with all that homology, can you still conquer it with short reads or long reads? Then we get into failed segments. So where did your assay not have enough coverage? Where are you not guaranteeing you got the sensitivity for your patient? And then are you going to Sanger rescue it, or are you going to report it back? But just knowing where those segments are in your assay. And then if you're in the carrier screening space, we get asked a lot to do pathogenic and likely pathogenic filtering. Nobody wants to go confirm some variant that's going to turn out to be a VUS and not end up on the report. So we get asked a lot to help filter down all of those variants into a smaller subset. So again, we can get the throughput that we need. And then we get asked a lot of questions about confirm bypass. I know the last couple talks focused on machine learning, which is what we started using a lot of to say, when is that variant good enough that you don't need to confirm it any further? So really taking it to that next level. And then you may also think about algorithms. That's what I thought of when I first said I work in bioinformatics. What kind of algorithms did I make, you ask? qPCR, digital PCR. We did a bunch of fragment analysis stuff for FRAX. We do Sanger, MLPA, and digital MLPA. We started making algorithms for whole exome and whole genome CNVs. Again, recurring theme, so long of my life. How do we organize all those testing workflows I just rattled off and all the algorithms within them? We use Sapio in a couple of our sites for a lot of our NGS and molecular testing. Not only do I love color, so I love this slide, but this is a collection of all the different workflows that we have built to date within the LIMS. It covers all of those different boxes I taught you in the beginning about that life of the specimen. So everything from intake review to informed consent through all those wet lab flows to all the different flavors of result processing that you can see on the right side of the screen all the way through to reporting. So a full rainbow of Qs. They're all built like Legos. You get to choose your Legos that you want to shove together to make your new assay pipeline. So reusability and site standardization, huge key things. I'll reiterate those in the end. Getting your sites to standardize is hard. The dream of standardization is wonderful, but pulling it off means you really get to quickly spin up new tests with less tickets, and that's definitely where we're all trying to be. I can act like we do everything great, but that would be a lie. So I figured, why not do a talk where I tell you where we didn't do a good job? You may look at this and think that that's intuitive, and that's great, but I only had it with hindsight, as did a room full of other people. So you build a system and you think you love it, and then you give it to your users, and then they tell you why they don't love it. You really have to get some thick skin. Where did they not like it? We made this action for the GCs where they have to go ahead and assign cases to themselves. That sounds simple until you assign the 2000th case to yourself and you don't like us anymore. And then it also became when you wanted to complete specimens, you had to click on that specimen and click complete. Sounded great, one button click. Again, not after you've done it 10,000 times. So it worked great for a few months, and then they came back to us and they were like, this is unsustainable. We're actually gonna have to hire more FTEs to click. Not happening. So come in, we all sat down with the business, informatics, and IT all sat together, and we said, how can we solve this? So our developers within Sapio created 2 different solutions. One is that we automatically assign cases overnight. We put in a little bit of config that said when folks are working and when they're not. Overnight, the scheduler comes in. Oh, you have 200 new cases in that queue. Let me go ahead and give 50 cases to each of the 4 GCs. I lied, there's like dozens of GCs, but it was a great round number to do. Now nobody has to click. You wake up in the morning, everything's assigned. The supervisor doesn't have to do it. You can input when people are out on vacation. You can still reassign them, but now you're not clicking for yourself anymore, and you get a much better idea about caseload rate when you roll in the morning. The other one I talked to you about is when they're done doing intake review, we were making them click a button that said complete, and then they pointed out that that's just inefficient. So now we gave them a screen where they can swipe a whole bunch of specimens at once, and then they can still just click complete. At least 5 of you in this room are like, you should have done it that way from the start. We were too busy building all of the other Legos, and again, you just don't realize it until you live it. So the big part about it is just acknowledge from the start that you're not going to get it right the first time. Somebody's going to outgrow whatever you created, and the trick is to be agile and quickly respond to their needs, and then they'll still love your system. And that's what happened here, right? Within a couple of months, we had rolled everything back out, and they were much more efficient than they were. The other one that came up, and the pandemic really brought this to the forefront, is alternate work location. Some people might choose clip art quickly. I spend a lot of time on it. It took me a long time to find a picture of somebody where you couldn't tell what kind of pants they were wearing. She could be in any kind of pants. I'm going to say yoga pants because that's usually me. Today I took a dress out just for Margaret. But the point is that now you're reviewing cases from home. You're not sitting in that CLIA-approved lab anymore, and our reports need to reflect where all of those components were performed. But you're not in your yoga pants every day, right? Someone goes to the office on Tuesday, but then they don't go in on Wednesday. So we needed that flexibility in order to capture where you were all the time in order to meet those CLIA needs. So during the pandemic, they allowed people to sign this out from home. And then that left this huge IT burden of we are preventing people from working from home until we can know that you're signing out from home. This really became important for retention. When everybody else was getting to go remote and our workers couldn't, and then really about filling 2nd and 3rd shift. Again, high throughput lab, we run 24/7. Someone is much more likely to take 3rd shift if they can do it from their home. So the solution, again, was through custom code. We give everybody this pop-up when they start their day that's basically like, where are you? And that's just because I don't know where you are by your computer. You can take it anywhere. So that way people can say, yes, I'm at the primary CLIA testing site, or no, I'm at this alternate CLIA site, because as some of you may know, you can register your house as a CLIA site. Or maybe you're just at a non-CLIA site, meaning your house that's not registered, and that's okay too. And then we can limit what functionality that user has by their work site to make sure people aren't signing out reports if they're not sitting in a CLIA space. So again, may seem intuitive in hindsight, but just not something that we had captured in our initial requirements, but now the LIMS empowers those users to get to stay from work from home if they want to, in whatever kind of pants you choose. Once you get through the pipeline, then it became, how do you know what went through the pipeline? I get asked questions like all the time. What kind of questions do I get asked? How many tests did we run in this given period? What's the breakdown of those test codes in that period? Or do you know how many samples were negative that we ran this January? I really do get asked these questions like I'm just supposed to know the answer right away. So it's really interesting considering we run hundreds of thousands of tests a day. I get asked a lot of questions about positive reports. Was it a positive or a VUS? Or what was the breakdown of that? Can you break that down by gene and by specimen type? However which way you get asked. How many assays failed last month? Did you see a spike in repeat rate? Or what % of variants were actually confirmed with a given technology, right? Maybe there's some algorithm problem where you're losing specificity. So the thing to do is self-empower your user to answer these questions so that you don't get an email. I get a lot of emails. I'm really trying to cut those down. So within Sapio, we went ahead and built out management reports. For those of you that aren't familiar with them, you get to put your cute little queries up at the top. You get to choose what those are. So for example, for this one, it's a to and from date, a group of test codes, and you can put in certain report types if you want. Obviously, as I've shown here, you can put in multiple. And then what it does is it gives you a cute little table in the bottom that gives you a button to export to Excel, because everybody loves Excel. But it's all about pictures, man. So then you can take that same table, and then the graphs are there for the users too. So they can see the breakdown of tests and the time period I gave. It's all fake data here, folks, sorry. And then you can also see how many of those were positive or negative or VUS by all of those test codes. So now the director doesn't have to email bioinformatics or a LIMS person. They can just go in and run their management report. They can even get cute little graphs that they can put in their own slide decks for their leaders. So we really thought it was nice. You can slice and dice it by site now that we run the same tests at different sites. So a really nice feature within. So I showed my group this in bioinformatics, and this is on the LIMS side, and I kind of straddle the line, and I was like, we got to make cool graphs like this. And they're like, you're right. And then this goes back to box plot April. And I was like, how about we make something that makes these box plots? I keep getting asked, right? So we had some lab troubles with a reagent that changed how it was stored. Again, hindsight. All you see is that your assay starts tanking, and you have no idea why, right? And you're really trying to trend that. So for those Python Shiny users in the room, welcome to the latest bioinformatics dashboard. Even Margaret hasn't seen this one yet. And we built it so that the lab could trend all their QC metrics, not only what's in the LIMS, but what they gathered at the bench, down to when they swapped to a new reagent lot, or when they used Hamilton 3 instead of Hamilton 2 to do that pipetting. Or for those of you that have, sequence a lot, there's always that sequencer that's a lemon, like you just know it's always going to create a problem. Now you can filter your data this way. So we gave everybody on the left-hand side, they can pick their location. So obviously I showed you QA data, but I picked just Westboro. We run different sequencer types. So you can toggle NovaSeq to NextSeq, for example. NovaSeq runs our exome data. You can pick your date range, and then you can pick a series of QC metrics. I'm obsessed with Fold 80 lately. For those that don't know, it just measures coverage uniformity. I've also learned that it's your canary in the coal mine if your assay starts to go down. So we went ahead, and then you can see all of your flow cells along the x-axis. Now you get your box plots for free. I'm not making them. You can still see all of your outliers. They can uncheck and check the flow cells to the right so they can throw out a bad run. And now they can trend and see how Fold 80 is looking. This is one of the metrics that started to look bad before people noticed, and we just didn't catch it until it fell off a cliff. So now they can trend it. If you haven't noticed yet, I love colors. So we introduced some happiness into everybody's world. And then right after we rolled it out, like 2 days later, someone was like, I really would like some correlation plots. I want to see Fold 80 versus AT dropout, because those are usually pretty correlated. So now I'm back to making manual scatters after box plots. So then we went ahead and evolved quickly. And by quickly, I mean within the next day, we rolled out the new plots. So again, Python Shiny, shout out there. And then they were like, I also want to plot by pool, because we pool 12 samples together into 8 pools across our 96 well plate, and then we pool 2 96 well plates together on a flow cell, again, high throughput. And then sometimes you just want to see pool performance. So in this plot, now you have AT dropout on your y-axis, low is good, and then Fold 80 on your x, again, low is good. And you can start to see, in plate 1, pools 4 and 5 look great. And pool 3 is just not playing nicely today. So this allows the lab to break it down not only by a plate level, but by a pool level. Now if a certain thermocycler or condition or pipette tip wasn't behaving well, you might be able to drill down into that pretty easily. The biggest thing is that the users just go to a website and they click whatever they want. They don't have to come to me. They can go to prod, they can go to QA, they can slice and dice it by sequencer and site, and then we've layered it on for different assays as well. So whether you're doing NIPS or targeted NGS, or you're in the somatic space and you're doing myeloid, everybody gets their little Python Shiny dashboard now as of just a couple months ago. And it really freed up my team from a lot of the data mining and plotting we were doing. And now the labs are catching their problems even faster. So really nice story there. So how does it all come together? If I look back into the last 18 months, which is when we started running all of our assays in a consolidated space, we've now run over 200,000 tests. Again, this is just in this LIMS. It's a small number for Labcorp, but it's big because it was my whole life for 3 years. We do NGS, qPCR, fragment analysis for FRAX, and all those confirms. There's 501 users across the 4 different sites that are in there, and that includes all the remote workers as well. 75 different APIs that connect to legacy systems, because we have a lot of different legacy LIMS in our space, as well as any internal databases and external tools that we loop. And just in this one sector, 550 flow cells and counting. So just to give everybody a little peek at the numbers over the last just shy of 18 months. How did we build all of that? Because I get asked that question a lot. It's a lot of JIRA tickets, 4,260. I have a t-shirt that says, if it's not in JIRA, it didn't happen. So it doesn't matter how small of a fix you want. If it's not in JIRA, I'm not changing that color for you. It took us 29 sprints to do those tickets, 28 different releases, 42 total epics. Again, all those Legos and work queues I showed you in the beginning. Who's we? 12 different LIMS developers, 10 bioinformaticians on my team, and 9 QA and UA testers, pretty much 3 years of their life growing it. We did one small assay and then just kept building on top of it. So please don't think we built all of this at once, right? You start small and then you just keep adding on once users realize they like you. So how do you want to bring this home and do it in your site as well? Here's my recipe for success. I didn't patent it, but I really do think it will get you there. The first is strategic partnerships between not only your LIMS and your bioinformatics team, but also your business users. If you're in the clinical space, if you don't get everybody in the same room, it's just going to cost you a lot of emails and change requests. So just pull them all together early, make sure everybody's made friends, get some team chats going with just those groups. It really will save you a lot. And then really continuing fostering cross-functional collaborations to make sure you're building reusable components. If I'm building something for extraction and I don't talk to the 4 sites that are going to use it, I risk having to build different flavors of extraction for every site. So really taking that partnership that you made in the first bullet and then using it to drive that collaboration and standardization is going to allow you to really get to that reusable Lego model that we all want. We do a lot of live demos. I used to do them every other week. I think it really helped with my public speaking skills, but you can let me know later. Leadership got to know what we were doing every 2 weeks. Users got to see it. They got to give us feedback in real time. You either heard oohs and ahs and you celebrated that night, or there was like dead silence and you knew you didn't nail it. But either way, you got that feedback every other week. Again, back to the Jira, though, I don't care how you do it as long as you're organized. If it's a sea of Post-it notes, you do you, but capture all those new thoughts and enhancements. When someone gives you that random idea in a call, don't dismiss it or forget to write it down. Sometimes those are the next big idea. We also poll our users pretty regularly for feedback. We have sessions where we drag that feedback out of them. You're only really ever going to get criticism most of the time. So if you want to have an uplifting conversation, corner them, bring free snacks, and then really have them. And I mean all the way down to your lab tech, right? Because they're the ones that are usually in your systems or touching your data or affecting the performance of whatever you're touching in your data lake. There's always going to be new technology. You're never going to get it right first try. Hopefully that's one thing you remember from this talk. Nobody's perfect. There's going to be a new exception or variant type or algorithm coming out there, and you just need to make sure you can respond to it quickly. And then the last thing, because again, so much plotting and dashboarding this year, leverage all the trending tools at your disposal. There's so many things out of the box. For those of you that are in Sapio, there's so much that is in that system already, but really making sure you're trending not only the performance of your data, but the performance of your LIMS, right, in terms of how fast it's responding along with all of your others. So there's so much more that goes into it, but hopefully this gave you a sneak peek into how we really do high throughput at Labcorp, and I'll definitely be around at lunch if there's any other questions. Thank you.
Any questions for Angela? I have a question. What are you doing with all your free time now?
No more box plot, yeah. Now we're expanding the dashboard into HIV typing and microbiome assays, right? So every assay has different data points that they really want to look at. So taking myself out of my just tiny NGS space and really kind of growing it into other sites that I don't interact with all the time has been our most recent endeavor.
I know you're a clinical lab, but you don't work against GXPs, right? You work since you're doing this agilely. Do you make, and I would assume that you have testing embedded after each sprint to ensure that the data is, I guess the data is good, right? Or the configuration will suit whatever requirements are needed, correct?
Yeah, and there's different layers to it, right? In bioinformatics, we do unit tests within the code. We started using PyTest for more automation. So on code check-in, here are these test cases that you should invoke. And then, we also moved to doing automated weekly regressions in our testing environment, where we're just dropping exomes and genomes and NextSeq runs every week and comparing the results back to the week before, and then highlighting any discrepancies that come up. So there might be changes over time that are intended, but just documenting those and really locking in versioning kind of really helped us become more agile.
And what was the length of your sprint?
These sprints were 2-week sprints. We started migrating to 3-week sprints now that we're in a good groove. But we started at 2 to really make sure users were seeing incremental improvements across the month. But once you've stabilized, I think 3 months allows your testers to just have more room to breathe.
I think this is an amazing story, by the way.
There's not a lot of science, but if you don't have this, you can't process your science. So.
Awesome work. Quick question for you. You mentioned you guys have a lot of legacy LIMS also that you have to go back and I can imagine that over time, over many years, the sensitivity and the specificity of the assay can improve. So when you go back in time to compare, I'm not sure how you guys are internally perhaps reflecting that in your dashboards as time passes when you do pooling and whatnot. Okay.
Yeah, sometimes things run under different test codes for us. So once something changes technology, it gets a new code. So we can use test code to segregate that performance out a lot. The other tricky thing is our region of interest changes over time, right? What we used to target for the assay 20 years ago may not be the same region we report now. So filling in that gap is something that you want to keep in mind too as you're aggregating all of it into a data lake. We're kind of more careful that as we're pulling it from those legacy LIMS, we're tagging it appropriately so that we know, hey, this data set's not going to have these 5 data points because it came from 30 years ago. So there's over 30 legacy LIMS right now that we're juggling, and a lot of them are getting consolidated under Sapio, which is nice. Less places for me to query my data from.
How much time does it take for the users to actually trust the process, right? Because it takes a lot of their time to go through user acceptance testing after every sprint. And how long did it take? And I'm sure it was probably a matter of a couple of months, maybe.
Yeah, I'd say so. We started with a small volume assay, 1,000 samples a month, and really let everybody get in there with the basic workflow. So I'd say within a few months, we were confident we nailed that. But stepping up to the exome genome level and the throughput that we were asked to do is really where, it took almost 2 years of development. With them doing UA right with us, the biggest thing were those live demos. I was taking samples end to end every week, clicking the same buttons you were going to click as the user. So that's really what got the most user faith early on, was being transparent and showing them, and then, ooh, I got and just owning that and showing them how to recover. So I think doing those live demos where it was just the raw experience really helped. And then once we crossed 100,000 samples, that's where everybody took a step back to say, okay, we're at multiple sites now. We've done 100,000 reports. Now, how do we make us be more efficient in each bottlenecked area?